ears to hear your presentation. Thank you.
Okay, thank you. Thank you for the very kind introduction. Can you hear me okay still?
Yes, we do hear you.
Okay. It's a pleasure to join you. So thank you again for the invitation. So I'm going to talk about work, and I just want to quickly thank some of the key people involved in the work, including Ali Mohabi, Wei Wei, and Tim Kraus, and I'll thank some others as we go along. So I'm going to tell you 3 short stories about dopamine. And the first one is called A Spectrum of Timescales. And this story begins a long time ago in the 1950s with the discovery that there are certain places in the rat brain that you can tickle with electrical current. And if you hook those places up to a lever, then rats will learn to press the lever to stimulate their own brains. And they'll become highly motivated to press the lever again and again and again. And when people started doing pretty exhaustive mappings of where in the brain this works, they found that many places work, but a lot of the places that work best were clustered along the mesolimbic dopamine pathway. And this result, and many other results, led to the idea that we have a reward pathway in the brain, going from these midbrain dopamine cells, especially the ventral tegmental area ones, to the striatum, especially the ventral part of the striatum called the nucleus accumbens. But when we talk about a reward pathway and dopamine and reward in general, what exactly do we mean by that? And I want to just refer to a very nice review a number of years ago by my former Michigan colleagues, Ken Berge and Terry Robinson, where they distinguished between 3 different meanings of the word reward. And one of the simplest meanings that we use in everyday life is the idea of pleasure or hedonics, liking things. Right? So that's one sense. Another sense is learning from rewards. Right? So if you perform an action and you get a reward, you're more likely to do the action again. So this is the idea of reinforcement. And a third idea is related to motivation. It's the idea that rewards are things that you want and are motivated to go and get. Right? So when we talk about dopamine reward, which of these things are we talking about? And in some very nice work, people exploited the fact that animals, including humans, have these very strongly conserved across evolution reactions to things that they like and things they don't like. So this is supposed to be a newborn baby with sugar in its mouth, or a non-human primate, or a rat. All of these react in a similar way with oral facial reactions when you put nice sweet stuff in their mouth, and in a different way when you put nasty stuff in their mouth. And so when you use this as a readout of whether animals like things, what you find is you don't need dopamine at all to like things. You can totally remove dopamine, especially even from the ventral striatum, and you still get these same liking reactions. The difference is that a rat that doesn't have dopamine in the ventral striatum won't work to obtain these rewards. All right. So we can try and, at least for now, remove this idea of pleasure from the concept of reward and dopamine and focus on some of these other things. All right. So as years have gone on, this idea of learning and reward has moved to especially focus on this reinforcement concept. And you already heard, I believe, yesterday from Wolfram Schultz and his famous work on reward prediction errors. And I'll just spend a few seconds to remind you that the sort of seminal findings here was that if you squirt fruit juice into a monkey's mouth and it's not expecting the fruit juice, then you get a burst of firing of dopamine cells. But if you make that fruit juice predictable by preceding it consistently with a cue at a fixed time ahead of the reward, then you get a burst. The burst transfers to the cue instead of the reward, even though the monkey still likes the juice and still licks avidly for the juice. All right. And that's what I just said. Okay, and you can push it back even further in time if you make that cue predictable by another cue. All right. So this is the very common concept in the field that dopamine cells signal reward prediction errors to help learning of reward predictions, right? And I'm going to refer to reward predictions as values. Okay, another way of thinking about this sort of theoretical conceptual framework, which has been extremely influential and is very powerful of reinforcement learning, is that the system is designed to generate these reward predictions or values and use those predictions to guide choices and motivation to work. And then as you go through life, you get feedback about those choices, and that feedback takes the form of these reward prediction errors, reward, RPEs. And the point of those RPEs is to feedback and update your reward predictions, right? So in this framework, it's important to emphasize that that's what reward prediction errors are. They're learning signals and nothing else. Another concept I have to just briefly update you on, remind you of, is the idea of temporal discounting. And this is pretty straightforward. If you have learned that you have to pass through a sequence of unrewarded states to get to a reward, then it seems pretty intuitive that it's better to be in a state that's close to reward, like number 9 here, than a state that's further away from the reward, number 2. So we can think of these values, how good it is to be in a state, as the time discounted distance from the future reward, right? And that's what we mean by values through most of this talk. It's these temporally discounted predicted future rewards. And once again, the point of a reinforcement learning algorithm is to use your ongoing experience to update this set of values. And the set of values together we'll call the value function. Okay. And one more connection here is that in the idea of reinforcement learning and reward prediction errors, we often talk about temporal difference reward prediction errors, which is the change from one moment to the next in your reward expectation, producing an error signal. So for example, if you're beginning work and you have to pass through a sequence to get reward, and then you find out halfway through that the reward is definitely coming, then you have a jump up in your reward predictions, and that jump up is a temporal difference reward prediction error. Okay. All right, and this one reason why this framework is so influential is that it actually works in the sense that it's been very effective in machine learning to make artificial agents that can do amazing things. And some examples here are just for playing Atari video games or playing Go at superhuman levels. And of course, there are many more examples in the last few years. All right, so let's talk about dopamine and reward prediction errors. And Schultz and others started elaborating on this idea now 25 years ago. They really were talking about a global broadcast signal that would go from midbrain and be sent throughout frontal cortex and striatal structures. And this fits very well with the idea of a reinforcement learning signal where you have this simple scalar signal. All right, but now, especially in the last few years, we have many reports that not all dopamine cells are doing the same thing. And so how should we think about this? So one way that people think about it is they're just different dopamine systems with different functions. So maybe some do reward, some do movement, some do salience. That's one perspective. I'm going to present a different perspective, which isn't to me, this perspective is not to say the other perspectives are wrong. It's just, I think, a useful perspective. So I need to just remind you of some of the anatomy here. So this is rat anatomy. We heard some very nice work from Peter on non-human primate anatomy in the last talk. I'm going to talk about rats. And within the striatum of the rat, the dorsolateral striatum here is a sensory motor striatum. If we go to this upper purple zone here, we get to dorsal medial striatum, which is more associative, cognitive area. And then down here, the nucleus accumbens is ventral striatum. It's supposed to be more motivational and emotional. All right. And the first piece of data I'm going to show you is extremely simple. We're going to use the dopamine optical sensor D-Light to measure dopamine fluctuations in real time. And we're just going to have a rat in a box. And every now and then, without warning, we're just going to deliver a sugar pellet reward and see what happens. And if I can go here, so this is work by Ali Mohabi in my lab. And here he's simultaneously measuring these dopamine fluctuations in these three different striatal subregions. And I hope what you can see is these fluctuations, and you can immediately see that there's more stuff happening in the dorsolateral striatum, intermediate DMS, and then when we play a click, you can see a jump up in all three signals, right? So some aspects of the signal, dopamine signal, are shared between these different subregions, but other regions are quite distinct. Other events are quite distinct between these regions. Okay. And I'm going to assume that you can still hear me even though I can't see you. because I'm in shared screen mode, but please interrupt me if that's not the case. All right, so I just showed you this example of these different dopamine signals in different places, and just simply plotting autocorrelograms or something, you can quantify how much faster the signals are changing in dorsolateral striatum compared to dorsal medial, and faster in dorsal medial compared to ventral striatum. All right, all the areas respond to an unexpected reward delivery at click, but they respond more briefly in dorsolateral striatum. Okay. So what happens in the context of a behavioral task? And I'm going to start by talking about this simple Pavlovian approach task where occasionally, as before, we just deliver unpredicted food pellet. And then sometimes we precede that with different tone pip trains. Pip, 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 pip. And depending on the pitch of the tone, it could be either a 75% chance of being followed by reward, 25%, or 0. And over the course of learning, we're just going to mix up these trial types, and we're going to train for 15 days in this case. And over the course of learning, animals learn to approach the food dispenser in advance of the reward, and they do so more for the 75% than for the 25%, and then more for the 25% than for the 0. And this is our behavioral measure of learning in this task. Okay, so what's happening with dopamine here? So if we look at these 3 areas in the dopamine signal, so this is what I showed you before. This is now the average signal in response to this unpredicted food response. If we look at dorsal medial striatum, then what we see resembles fairly well a sort of reward prediction error idea, that you see a strong response to the 75% cue, a small response to the 25% cue, and actually a negative response by the end of training to the 0% cue. So this, so far, so good. If we look at ventral striatum, we see kind of the same thing, but it's weaker, right? The difference between these signals is less apparent, and actually, even after 15 days of training, still stays slightly positive for the 0% cue. So yes, just seems slower at discriminating between these cues. And I should emphasize, this is in the same rats, so it's not just a difference in behavior. And if we look at dorsolateral striatum, we see something a bit different, which is that, even after extended training, the response to this, high probability of reward cue remains very small. So what's going on here? So are these just different signals, or can we find ways to think about them that unifies these things together? So this is the same data I was just showing you in this weak response to the 75% cue. But it's not simply that the dorsolateral striatum doesn't care about this cue, because if you precede the reward with this cue and make it predictable, then the response is much smaller than if the animal wasn't expecting it, right? So it does look like a reward prediction error signal, but just a different one in dorsolateral and dorsal medial striatum. So how is it different? Okay, and we've been playing with the idea that maybe it's a reward prediction error signal in each case, but that the underlying values, the underlying reward predictions are different. And how are they different, right? So the story really depends on this idea of temporal discounting that I introduced before, right? So if you discount the future at different rates, you could imagine that even with a smaller interval to reward, that you would have a smaller response to cue. All right, and we can quantify this and model this in various ways. We started out by using a very simplistic but a classic model in the dopamine field, where you just break up this interval between cues and rewards into lots of little states and go from there. All right, and when you do that, you feed the animal's actual experience into the model, what you can see is that, yes, you have this different discount rates on value that produces different reward prediction errors, and the response to the cue is much smaller when you're discounting the future at faster rates. And this is not terribly surprising. Okay, so this simple model can explain why the dorsolateral striatal responses are weak to 75% cue. And to reiterate why that is, the cue is predicting a reward that's just too far away for the dorsolateral striatum to care, given how fast it's discounting the future. Right? But it doesn't explain everything about our data, And in particular, this model would predict a really robust response in the ventral striatum if it was just discounting more slowly, and that's not what we see. All right. So we can't explain with this simple model the poor discrimination by the ventral striatum. But these models are too simplistic, and they only model the interval between the cue and the reward. And of course, that's not the full experience that the animal is having. So rat experience is not really trial-based, but really continuous in time. So this is just a cartoon saying in one of these sessions, we had many, many trials, 120 trials or so. And if you were discounting the future very fast, then even the interval within a single trial might be too long, right, for you to care. But if you're incorporating a much wider time horizon, then you might have a different effect. And so we can model that in this way. We use recurrent neural networks with 3 subnetworks with different discount rates. And we fed this continuous set of experiences into the model. And when we do that, and I should emphasize that instead of breaking up time into these sort of individuals, substates, we represented time implicitly using the network dynamics. All right. And this is on bioRxiv if you're interested in the details. And when we do that, we can recapitulate many detailed features of dopamine release if you make the assumption for now that dopamine pulses reflect reward prediction errors. This is just this here. All right. I'm going to skip over and explain it this way. All right. So just to reiterate what I said, the dorsolateral striatum is reflecting predictions on short time scales, right? And this is important because the way we set up the task is we have these different cues, and the cues are only informative about reward a few seconds later, right? So that's what the cues are doing in this task, right? That was a choice. If we look at dorsolateral striatum, this time scale of a single trial is too long for the dorsolateral striatum to be keeping track of this properly, right? Conversely, the ventral striatum dopamine seems to reflect these underlying values that are keeping track of much longer time horizons. And on those longer time horizons, all the cues aren't so different to each other, right? You may get this cue just followed by a reward, but it's also followed by a reward here and a reward here. And all of the cues over the time scale of a few minutes are followed by a bunch of rewards. So it's harder for the system to discriminate. Whereas the dorsal medial striatum reflects learned predictions over a time scale that matches the structure of the task that we set up, right? And so that's why we would argue we see these clear reward prediction error responses. Okay, so why on earth should it be like this, right? So you probably are aware that there is many parallel loop circuits, cortex through the basal ganglia and thalamus and back to cortex. And there's lots of lines of evidence that these are involved in organizing different aspects of behavior that have different intrinsic timescales, right? So if I'm learning to sing a song and I sing the note better than I thought, right? What matters was the state of my vocal cords, a fraction of a second earlier. So the feedback is fast, right? It doesn't matter what I was singing, 2 minutes ago. Whereas, many other behaviors that we do require extended time to decide whether they were a good idea or not. If I have to decide whether to go hunt the mammoth in this valley or that valley, I may not know whether I made the right choice for a day or more. Right? So we have to organize our behavior over different timescales. And our hypothesis is that all these loop circuits are involved in invigorating, motivating behavior and learning what to do based on these reward predictions, but just operating over different time horizons, right? And that's the idea there. And one other interesting aspect of this is that there are some very fascinating observations for many years about apparent inconsistencies in human and animal choice behaviors, right? So as rewards get nearer, we seem to be increasingly impatient. And this results in a pattern of behavior that's famously called hyperbolic discounting. And there's been some very interesting suggestions in the field that this hyperbolic discounting, which seems on one sense irrational, may actually be reflecting the collective behavior of multiple loop circuits, multiple subagents, each of which is discounting the future over different timescales. Okay. So that was the end of the first story. I'm just going to quit to make sure you can still, I'm assuming you can still hear me. So I'll tell the second story. Okay, so this is called local sculpting of value signals. Okay, so this story begins with talking about Parkinson's disease. So as I'm sure you're all aware, Parkinson's disease is caused in large part by, or the symptoms are caused in large part by loss of dopamine cells in the midbrain, right? And is characterized by this smaller, slower movements and many other features, right? And, you know, Parkinson's disease is normally referred to as a movement disorder, but it's not exactly right, you know? And there's a very nice paper here and others arguing that the way we should think about Parkinson's disease is not simply as an inability to move, but as a failure of implicit motivation, that movements are deemed, not consciously necessarily, but deemed less worthwhile. And so more generally, I argue that basal ganglia circuits are important for deciding how to allocate limited resources that are available to you. And these resources could be attention, effort, time, based on what's worthwhile. And these may reflect the functions of different subcircuits through the basal ganglia. Right? And another line of evidence, more lines of evidence that dopamine isn't just a learning signal, comes from work like this, where you can mess with dopamine in various ways, either genetically or by giving drugs, stimulant drugs. If we take cocaine, hypothetically, we can work harder, and it seems to be linked to motivation and effort, but not so much to learning. Okay, and I've showed you this slide before, and the idea, the issue here is that in the dominant theory of dopamine function, we have these reward prediction errors, but they're really learning signals, and they're not supposed to be involved in this other half of the cycle in motivating and choosing. Okay. I just want to give a little bit more of the evidence that dopamine, especially in the ventral striatum, is important for the sort of motivated decision making. And of course, there's many, many lines of evidence. Here's a couple of nice pieces of work from John Salomon's group showing, first of all, that if you have a rat and you remove dopamine from the ventral striatum and you ask it to push a lever for reward, well, if the rat just has to push the lever one time to get the reward, then it shows no deficits at all. But if it has to push the lever more and more for reward, then the rats give up more easily, right? Compared to a normal rat that'll keep on working and working, right? Here's another example. If you have a tea maze and the rat has to choose between one sugar pellet, which is just right there and available, or two sugar pellets for which the rat has to climb over a barrier to get them, a normal rat will decide it's worth going over here to collect the two pellets. But a rat with a lesion of dopamine in the ventral striatum will just go over and get the one pellet. Right? So there's a lot of evidence for ventral striatal dopamine in particular in a sort of flexible approach choices in which you have to engage a sequence of actions that may lead eventually to reward. Okay, but what hasn't been so clear is how this function is reflected in the dynamics of dopamine. All right. And one scheme, this is from Wolfram Schultz a number of years ago, one suggestion was that maybe these fast pulses in dopamine, the kind of stuff I was showing you earlier in the talk, does these learning signals, these reward prediction errors. Whereas there's a slower, more sustained changes, we might call tonic dopamine, that does motivation in some sense. Right? So that's one thing we're going to explore in a minute. And another aspect that's become again, a topic of discussion in the field recently is maybe there's yet another time scale where you just need some dopamine around. It doesn't have to change at all, but just some dopamine is needed to enable you to not have Parkinson's disease. It's an interesting idea. It's on current further discussion. Okay, and then talking about this idea of slower changes, of tonic changes in dopamine. There's a very nice paper here from NIV, Nathaniel Daw and Peter Dayan, a number of years ago, so trying to provide a computational footing for this idea, suggesting that these slow changes in dopamine are signaling the rate of reward, right? So if you're getting rewarded a lot, you'd have some higher level of dopamine, and this might make you more motivated to continue performing some task. All right. Okay, so that's a lot of talking. Let me get back to some data. And a lot of the work I'm going to show in this middle story involves the rats in a box, and the rat can poke in these various holes, they can light up, and it can get sugar pellets from the other side. All right, and I'm going to talk about this trial and error task or a bandit task. And the way it works is this. At the beginning of a trial, one of the holes lights up, and then the rat can go and stick its nose in the hole, and it takes some time to do that. And we're going to call that time the latency. And as you'll see in a minute, we'll use this latency as our index of motivation in this task. Right? And once the rat has put its nose in the hole, it can wait there for a variable time until it hears a go cue, which is just white noise. And then it pulls out and sticks its nose into one of the side ports, either to the left or in this case to the right. And sometimes when it does that, it hears a click, which is the sound of a sugar pellet being delivered behind it, which it can then go and collect. And this is a probabilistically rewarded task, a bit like the ones Peter was talking about earlier today. And the probabilities change every now and then. So for example, in this beginning of this session, tall tick marks indicate a rewarded trial, short tick marks indicate unrewarded trials, and the initial probabilities were 50% for both left and right. And so the rat tried left, got a reward, tried left again, no reward, tried left again, no reward, tried right, no reward, and so forth. And this is just part of a session. A full session might look like this, a few hundred trials. And we're changing the nominal reward probabilities here. And you can probably see that when the reward probability is high for left, then the animal switches over to doing mostly lefts and vice versa. But it's not just the choices of the animal that are changing, that are adapting. It's also its motivation to play the game at all. So for example, here I'm just showing the same data, just forgetting about left and right, but just showing rewarded versus unrewarded. And when you have a higher density of rewards, the latency on this inverted scale here gets very short. So the animals are quick to initiate trials when they've been receiving lots of rewards. Okay, and we can see that here. This is the left-right choice aspect where they switch over to going to the better choice. And this is the vigor or motivation aspect where the latency gets short as the number of rewards they've received recently gets large. Right? So these are two different aspects of behavior in this task that the rats are adapting to their ongoing experience. Okay, so how should we think about this? So how do we connect this latencies to reward history? So one way of looking at this is as the probability that the rat will decide to engage in the task at each moment. So if the rat has got lots of rewards recently, then after the light comes on, there's a much higher chance that the rat will stop whatever else it's doing and choose to approach the light and engage. Okay, so the hazard rate, this probability of engaging, is higher when the reward rate is higher. Okay, this is all published stuff so far. So I've spent a bit of time talking about this behavioral task, and now we're in a position to look at how dopamine correlates with these reward rates and influences decisions. So first of all, let's just look at some optogenetics data. And this is an experiment in which Arif Hamid in my lab put channelrhodopsin into dopamine cells in the ventral tegmental area. You can stimulate these cells and measure using, in this case, voltammetry, the response in the ventral striatum. And when he does that, he's giving here a half-second train of pulses. He can produce a nice dopamine response. And what Arif did was he tweaked the parameters, the power and so forth, to not use a crazy amount of dopamine. In fact, to have an amount of dopamine that was comparable to what you see with the normal reward delivery. Right? And he's going to give this extra pulse of dopamine at different times in this task. He's either going to give it just after the side in. So when the animal finds out that it is or is not going to receive a reward, right? Or he's going to give it right at the start of the trial, at the same time as the light comes on. Right? And in each case, we're only presenting the laser on 30% of trials in different sessions these two times, right? And the idea here is that if the animal makes a choice and gets extra dopamine, is that reinforcing the choice? Does it make the animal more likely to choose the reward, make that choice again? But here it's saying, how does dopamine affect the animal's motivation to work in this task? Can it affect behavior right away? So first of all, this reinforcing action, yes, indeed, if the animal makes a choice and gets extra dopamine, he's more likely to make the same choice next time. And this doesn't work if you don't have Cre in the right place. And you can also push behavior the other way by inhibiting dopamine cells. Okay, so that's so far so good. This looks like a reinforcement signal consistent with this idea of dopamine, the signaling, reward prediction errors, and so forth. But we also see that when we give laser at the very beginning of the trial, we can immediately affect the latencies to approach. And if we express this as a hazard rate, here's our laser pulses, then within less than a second, what we're seeing is we're influencing the animal's choice to approach. Right? So this is, let me see, just say that again. So if the animal hasn't already decided to approach, giving extra dopamine makes the animal more likely to approach on the same trial, right, within just a few hundred milliseconds. I just want to emphasize this result because there's sort of ongoing controversy in the dopamine field about how quickly dopamine influences behavior. And this, I think, is one of the more straightforward to interpret examples where when the animal's primed to make this approach behavior, but isn't sure whether to do it based upon how motivated he is, giving extra dopamine makes him immediately more motivated. Okay, so as I just showed you, dopamine can affect both motivation, that's current behavior, and learning, the future behavior in this task. And this is already inconsistent with just a reward prediction error only account of dopamine. Again, because RPEs are learning signals. So that was an optogenetic experiment, but how did natural dopamine signals evolve during this task? All right, so we have measured dopamine in many ways. It's almost always painful, but I'm just going to show you some of the different ways we've done it. So with Bob Kennedy back in Michigan, we used his fancy version of microdialysis. This gives a 1-minute time resolution, which we might want to call tonic dopamine. And he measured not just dopamine, but working with him, we measured not just dopamine, but many other neurochemicals at the same time. Beginning with Brennan Aragona, we used fast-scan cyclic voltammetry, much faster resolution, focused on dopamine, tends to have some stability problems after a minute or two. And then beginning a few years ago, we worked with Lin Tian and Tommaso Patriarchi using their cool new D-light sensor, which I already showed you a bit of data from this before. Okay, so when we used microdialysis, this is work from Jeff Pettibone in my lab. He actually tried a number of different places in the forebrain, including the core of the nucleus accumbens, the shell, also mediostriatum, and then a range of these sort of medial forebrain area, medial frontal cortical areas. And if he just did a simple cross correlation between dopamine release measured on this minute-by-minute time scale and reward rate, then he saw this nice response in the core, not in the shell, not in dorsal medial striatum, and he also saw a response in the ventral part of the prelimbic cortex. And this was very specific to dopamine, right? So dopamine has this relationship to reward rate, and lots of other neurotransmitters do not, right? So far, this seems good. And if we map this out, we see these two hot spots that in the ventral medial PFC and in the accumbens core. And I find it cute. I'm not quite sure how much to believe this, but if you look at these meta-analyses of human fMRI studies, then these correspond nicely to two spots that consistently show up when you look for correlations with subjective value across different tasks. Okay. All right, so far this seems pretty good match to this idea of a slower tonic dopamine signal that correlates with reward rate, right? So blah, blah. And so as we switch from a block going from higher reward rates to lower reward rates, the latencies get slower and vice versa, and dopamine's going in the opposite direction. Right. So this seems consistent with this long-term reward rate signal, but how slow is the signal really? I mean, does this reflect the actual dynamics of dopamine, or is it reflecting simply the time resolution of our measurement technique? Okay. We'll come back to that in a second. And then there's also this question of if you see slower changes in dopamine, how are they coming about? And so this is a review from Tony Grace, arguing based on studies over many years in anesthetized animals, that there's different populations of dopamine cells, some of which are not active, and others can change their rate, and changes in the number or the firing rate of active dopamine cells can produce these tonic changes in dopamine. So Ali Maibi in my lab a number of years ago now started doing optogenetic identification of dopamine cells in the VTA in freely moving animals. And this is just an example. He has optic fiber with tetrodes around. He puts that in. They're expressing channel rhodopsin, and he can look for responses to these blue light pulses. And when he does that, he can identify in blue here these tagged dopamine cells and distinguish them from population cluster of cells that are clearly not dopamine cells. Okay. So what happens with this? So here, again, we have a session of our bandit task. The probabilities are changing. I already told you that the reward rate fluctuations in the overall reward rate sort of correlate pretty well with the changes in latency, the animal's motivation to respond. So what's happening with the firing rate of dopamine cells? The answer is absolutely nothing. All right. So even though this is a task where I showed you that there was changes on these longer timescales between dopamine release in the nucleus accumbens and motivation, reward rate, the dopamine cell, in this case at least, doesn't care at all about these changes in reward rate. And in fact, none of our VTA cells, dopamine or non-dopamine, cared about these changes in reward rate. Right? Oops. Sorry. So no effect of reward history on the average firing rate of dopamine cells or their burstiness or anything else that we could measure. And this is even true for dopamine cells that we confirmed are projecting directly to the nucleus accumbens through a retrograde tagging. Right? And I should just mention that there's sort of related results from the Uchida group in mice in blocks of trials where the animals were receiving more reward or fewer rewards. They also didn't see any changes in the firing rate of dopamine cells. Okay. So why is there a mismatch between tonic dopamine cell firing and quote unquote tonic dopamine cell release? I think, as I mentioned before, we now need to turn to higher resolution methods. So here, starting with voltammetry, you can see voltammetry signals and their responses to rewards and to emissions in this task. And when we look at these signals, this is just one session, for example, and we're now going to align the data on different events. This is the light coming on. This is center in. And then here's when the animal finds out it's going to get a reward. There's this click. And then actually in this task, we generally saw the highest increases in dopamine as the animal ran over to the food port to collect a reward. Okay. And just to go back to this again, I want to sort of point out this increase that's happening even before, just as the animal runs over to begin the task and then running over to collect the food. And we're not the first to see this kind of thing. This is from some of the earliest voltammetry studies in behaving animals from Paul Phillips, as the animal prepares to push a lever for cocaine or for food from Mitch Reutemann. And there's another example from Kate Wassom's lab. So these ramping up just before this, actions, motivated actions. And here's another example from Paul Phillips and Anne Grabiel in a maze task, with a simple maze task, with the animal running through a maze arm and dopamine ramping up as the animal gets closer and closer to reward. Okay. So now we can compare what's happening at the fast time scale, with dopamine cell firing and dopamine release. So let's do that here. So we find that the dopamine cells, these are identified dopamine cells, respond strongly to the onset of cues, relevant cues in this task, whether that's light on or the go cue or the reward cue at side in. But we don't see it sort of ramping up as the animal approaches the reward. Whereas if we look with a fast sensor, D-light sensor, we see this ramping up as the animal initiates trials and ramping up as the animal gets closer to reward. Okay, and we can see this most clearly perhaps if we just look at this set of trials where the animal doesn't respond immediately to the cue, light on cue, but takes a couple seconds to respond to it. So in those trials, if we align data on the light on or the reward, nose in to get the reward, nose in to begin the trial, then you see there's no increase here in the VTA dopamine cell firing, but there's a big increase in the D-light signal. We can also see the same signal with voltammetry. It's just a bit slower, lower time resolution technique. Okay. All right, so I just said that. There's rapid ramp in dopamine before center in. Okay. So this is, again, a controversial finding in the field. So some people have performed a number of studies and said, hey, in my study, dopamine self-firing and dopamine release look just the same. And we don't disagree with that because there are many situations in which they're very closely linked to each other, right? So for example, when you see the response to a reward in this task, right? So this is the reward click. We see this nice burst of dopamine self-firing, and this burst is stronger when the animal had less expectation that it was going to happen, right? So if you have a low rate of receiving rewards, and now you receive a reward, you see a strong phasic burst of dopamine self-firing. And within less than half a second, you're seeing a corresponding burst of dopamine release in the nucleus accumbens here, measured with D-light. That's also scaling with reward prediction errors. Right? So this feature seems to match up pretty nicely, right? But other features do not. So even before the reward cue here, the dopamine levels in the accumbens are tracking the reward rate, even though we don't see any difference in the dopamine self-firing. Okay. So how could this come about? How can we get a dissociation between self-firing and release? And we've known for a very long time, since the 80s, maybe even earlier actually, that there are mechanisms for controlling dopamine release locally within the striatum that don't require changes in dopamine cell firing. And one of the most prominent of these mechanisms involves cholinergic interneurons in the striatum. And acetylcholine and dopamine form these dense networks in the striatum. Dense varicosities are intertwined, and dopamine release varicosities have these nicotinic acetylcholine receptors that are strongly responsive to local acetylcholine release. And we've known for a number of years now that if you stimulate the cholinergic interneurons, then you can immediately get triggered dopamine release, even if the ventral tegmental area is offline. Right? And very interesting work from Pascal Kaiser's group seems to recently, this past year, show that this actually, the nicotinic receptor activation can trigger action potentials locally in these dopamine neuron axons. Okay, so in this experiment, Ali was tickling cholinergic neurons using channel rhodopsin, while simultaneously through the same optic fiber monitoring dopamine release using a red-shifted version of D-light. And what he found was that if you start stimulating the cholinergics, you can get an increase in dopamine release, and it tracks very nicely the time course of the stimulation. And this is in freely behaving animals. Right? And then complementary work from Val Collins in my lab used drug infusions into the nucleus accumbens core and showed that if you, first of all, in our bandit task, this is our latencies, this is remember our measure of motivation to work, that if you block dopamine in the accumbens core, rats are less willing to work, they perform fewer trials, and the trials they do perform have longer latencies. And we can get similar effects in a dose-dependent way if we block nicotinic receptors using this selective agonist DH-beta-E. So this is telling us that both dopamine and acetylcholine acting on nicotinic receptors are important for the motivation to work in this task. So can we observe dynamics of acetylcholine activity that could be responsible for driving dopamine release and motivating the animal to work? So this is the data I was showing you before, that dopamine cells are not increasing at the time that dopamine release is increasing. And Alios also found that using GCAMP in cholinergics, that you can see an increase in the activity of the cholinergic cells at the same times as we're getting these release ramps in dopamine. Okay, I just said that. So this is one of several lines of evidence that maybe the cholinergic interneurons are locally driving dopamine release to help contribute to motivated behavior. Okay, I think I went pretty fast over that, but I do want to tell one more story. This is called propagating value over space. I'll try and get through this. All right, so I wanted to quickly introduce or remind you of the credit assignment problem. And this problem goes as follows. Often we go through the world and we make choices and we get feedback, but often the consequences of our choices are separated in space and time from those choices themselves. And this separation can make it a challenge to learn. How do you know which choices were right if they were separated from the feedback is delayed? And a lot of algorithms that are developed in reinforcement learning are specifically designed to overcome this challenge. Right? And this is interesting because the tasks that neuroscientists tend to use, like me, tend to use to investigate dopamine tend to be very simple tasks. Left or right choices, or cue predicts reward, or in a maze, you have a left or a right choice, right? Whereas in computer science, the algorithms that are being used to develop the procedures, the tests that are used to test and develop algorithms tend to be complicated. And they have, meaning multiple steps to get to reward, for example. And they have to be complicated because otherwise you don't really have a credit assignment problem, right? So to get around this, or to try and investigate dopamine dynamics in a more naturalistic setting, last few years we've been moving back to maze tasks. But here is a slightly more elaborate maze. And hopefully you can see the video. This is a little fast. So there's a rat running between the corners of this big triangle. And at each corner, there's a probabilistic reward again. Okay. And just like in the bandit task, those probabilities can change every now and then. But another feature that's different here is that sometimes we pick up one of these barriers and move it so that the paths available to get to reward will change. Right? Okay. And the overall design is to have a task where the animal makes many left and right choices between each reward, and then has this challenge of knowing which of those choices to update to know what worked. Okay, so this is just some behavior just to show the blue here is the occupancy of different places, and these are the reward probabilities of the corners. If we change those reward probabilities, the animal adapts. So we make this port less good, the animal doesn't go there as much, pretty straightforward. But if we can also change the path, so here we have a barrier, and we're going to move this barrier just a little bit over from here to here. And now, but we haven't changed the reward probabilities. So even though we haven't changed reward probabilities, the animal is now less likely to come over here because the path to get there has become longer. Right? So animals are balancing out the benefits, the estimated reward probabilities, with the costs, that is the path lengths to get to places. Right? And I'm going to skip over a couple of things. So what happens with dopamine in this task? Well, a few things. One is that as the animal gets a reward, we see a reward prediction error coding, no surprise. It's nice to see that. But we also see error-like signals at other times. And so here's an example where either the animal encounters a path that's newly available. It was blocked before, but now it's open, or newly blocked. And when you see a newly available path, you see a big pulse in dopamine. When you encounter a newly blocked path, we don't see anything. So we're still trying to understand. This seems like some kind of error signal, but we're still trying to understand exactly what this means. The main thing we're studying in this task is these value-dependent ramps. So overall, as the animal rounds to a reward port, you see a ramping up in dopamine, and this ramp depends upon the port reward probability. If the animal is running towards a better port, the ramp is steeper. Okay, so this recapitulates a lot of the ramp during reward approach literature that previously exists. And we can use this to show a correspondence at the hex by hex, the individual location level, between the reward at each location and dopamine. So you're running along this path, the value of each of these hexes is increasing because you're getting closer to a reward, and dopamine's increasing too. And we can show a nice correspondence at the individual location level between these dopamine signals and discounted future reward. Okay, and this is adapting to the history that the animal is experiencing. So if the animal runs along a path and gets rewarded again and again, then dopamine ramp goes up. And if he fails to get rewarded again and again, it's ramping down. Okay. So how is this update taking place? All right. So in other words, what algorithms is the brain using to solve this problem of credit assignment? So we're going to start by testing out different candidate algorithms. And we're going to start here with some of the simplest ones. So let's start with, one second here. We'll start with these temporal difference learning algorithms. And the key thing here is that if you get a reward, then you update the value of the preceding state. So we can either allow the algorithm to update just the preceding state or to update the values of all the places that were passed through before. Okay, this will be called TD0. So when we have the animal run successive versions of the same path, what we see is, you go from a unrewarded to a rewarded trial, and the ramp bumps up, and it bumps up preferentially in this earlier part, in this part that's closer to reward. And if we give more and more unrewarded trials in a row, we can see what looks like a bump of dopamine passing backwards earlier in time. So this is a sort of key signature of this TD0 type algorithm, that the value signal depends preferentially on what happened on previous trials and earlier trials when you're further away from the reward. All right. So when we use regression analysis, we can show that more distant hexes from reward are actually more influenced by older outcomes than they were influenced by what happened on the last trial. So this is strong evidence, we feel, for temporal difference learning in this task. And we just put this work on bioRxiv if you're interested in taking a look at it. But is temporal difference learning enough? And I just want to quickly finish up with a couple slides suggesting that no, there's actually other stuff happening as well that's being used by the brain to update these value signals. So behaviorally, if we have a rat making a choice, then the rat is more likely to move to a port if it was rewarded on the last trial. But it turns out that that's true even if the rat was rewarded at that port from the alternative path. So this is interesting because in a simple temporal difference account of things, you only update values along trajectories that you actually experience, right? Whereas here, what seems to be happening, both in terms of the behavior and in terms of dopamine signals, is that the rat can infer that if it went this way to get a reward, and if it went this way and got a reward, then it's also better to go to the same reward location following an alternative path to get there. So there seems to be some kind of inference that the animal's using based upon its knowledge of the maze and its structure. Okay. And when we actually make models that use a combination of a local update of values, as in the temporal difference algorithm, together with a global update of values using knowledge, using a internal model of the maze, we can actually account for the dopamine signals at each location better than using either model alone. Okay. So how could this work? So just to reiterate, animals seem to use internal models to infer values, and these models this use of these models is reflected in dopamine signals. How does this work? And we're currently investigating sort of the possibility that the hippocampus and the hippocampus's ability to simulate or imagine different futures or pasts is essential for this model-based decision making. And this is a collaboration with Lowen Frank at UCSF and Nathaniel Dorn. And I just want to finish with the last couple slides on this. Here we're using very high density custom probes into the hippocampus, CA1, to record large ensembles of hippocampal neurons in this maze task. And when you do that, you can obviously record individual cells. Most people are probably familiar with the phenomenon of place cells in the hippocampus. These are just examples of cells that were active at specific locations within the maze. And I hope you can imagine that if you recorded many of these cells, hundreds of these simultaneously, then you could decode where the rat actually was. And this works extremely well most of the time. But sometimes the decoded location doesn't actually correspond to where the rat was. And I hope this video is going to come out, but we'll see. So in this video, I'm going to just show you slowed down a few seconds of decoded position. That's going to be the colors compared to the actual location, which is the green dot. So here, the rat is at one of these ports, and now it starts to move away. And you can see the decoded position is sweeping ahead of the rat and sort of sampling these different places that the rat could be going to. All right, so sort of sweeping ahead. And Lowen Frank has previous work showing this sort of alternating exploration of future trajectories within each theta cycle. Right. So here's the rat's at a reward port. And then occasionally what's going to also happen at the reward ports is that the decoded position jumps a long way to alternative locations within the maze. I'll just show a few more seconds of this because I know I'm short on time. So again, the rat's starting another trajectory through the maze, and the decoder location sweeps ahead across these alternative paths, and so forth. And maybe we'll get to see another jump at the end. And I apologize if over the long distance the video doesn't work as well as it would otherwise. Okay. All right, so what we'll see. Okay, and you can hopefully see that just for a fraction of a second, the decoded location jumped to some of these critical choice points over here. So what we're currently doing is combining this kind of decoding analysis together with dopamine and trying to see whether these non-local representations are being used for one or both of the following two functions. One is to look ahead and plan and evaluate possible futures. And the other is, having received reward, to look back and test out other places and other choices that could have been used to get to this recording location. These are two forms of model-based value update. Okay, so just to give you conclusions, I showed you a lot today, so thank you for your patience. I argued that striatal dopamine can signal both prediction errors and predictions, right? That subregion-specific dopamine dynamics reflect a spectrum of internal timescales for decision-making and value updating. That, especially in the ventral striatum, these value signals are involved in decisions to work, that is, to engage in a sequence of unrewarded activities in order to maybe get reward. And that ramps in dopamine release can occur without changes in dopamine cell firing, and instead they seem to be linked to changes in cholinergic interneuron activity. In a novel maze task, I showed you that ventral striatal dopamine encodes these values, spatially discounted values of different places within the maze, and that they are updated in two different ways. Temporal difference, like propagation of value through space over multiple trials, and all this sort of inference using internal models for all maze locations. And finally, we're currently examining whether this model-based inference uses hippocampal non-local representation to update values. There's a lot of stuff, so sorry about that. And once again, to thank the people involved in this work, especially Ali Moabi, Wei Wei, Tim Kraus, Arif Hamid, and thank you all for your patience in listening.
Okay, thank you so much, Josh, for the very interesting and full of data and insight talk. If there are any questions from the audience.
Hello, thank you for your great talk. It was awesome. I've got a
Thank you.
question. In the last part of your talk, you mentioned that the algorithm that govern the release of dopamine actually updates in a manner, and I wonder if it is possible that this update use a method like backpropagation that we have in artificial neural nets, and if it is possible that such our brain use such technique to update the value of algorithm, how much does it need to be trained in order to learn an optimal algorithm?
Yeah, I hope I understand the question. So if I understand the question, so you're showing evidence of different algorithms being involved in the learning process, and does the animal have to be trained to use these algorithms? And I think that my guess is that these are algorithms that are sort of hardwired into the architecture of the circuitry, and that this is why these circuits are set up the way they do, is to use these different algorithms. But I think it's an open question. It may be that there's some flexibility there in the structure of the algorithms being used. So for example, in the earlier part of the talk where we were talking about different timescales, a related question to what you're asking is, are these timescales sort of fixed for each circuit, reflecting the intrinsic properties of each circuit, or are they adapting to the statistics and properties of the task context? And we don't know the answer to that question. I think it may be some combination of both of those things.
Thank you for clarification. That was good.
Any other questions?
Hello, thank you for your
Hello.
presentation. My question is, as as you told, dopamine somehow makes the motivation. But my question is somehow about addiction. You know, we know in addiction the dopamine level goes very high, and this makes damage to dopamine cells. Uh, if dopamine cells get impaired in such a way, then how this brain algorithm could adapt and manage to rescue the the alive being from being not motivated in anything, because they don't release dopamine well, and they don't absorb dopamine well.
Okay. I heard part of your question. So I want to just argue with you for a little bit. So I think you said that in addiction that there was a big increases or decreases in dopamine. I actually don't think that's true. So I think at least for a long time, I've taken the position that addiction reflects specific learned patterns, like memories, right, rather than a sort of overall poisoning of the circuitry. So this is a long-standing debate in the field. So does a state of addiction reflect a sort of dysfunction of circuitry in the sense that it either releases too much dopamine, or it has a state of too much expression of some genes? Or an alternative perspective, just not necessarily opposed, but maybe complementary, is that when we get addicted to substances, we're not getting addicted in general. We're getting addicted to specific substances in specific contexts and specific developing specific habits and so forth. And that this is reflecting very strong formation of specific memories. So we think that the role of dopamine in learning is very strongly related to the ability of drugs of abuse to release dopamine, and that the excessive release and uncontrolled release of dopamine can promote these very strong memories. But we've been arguing that for many years. I think the second part of your question, I don't think I quite heard very well, but I hope that was at least somewhat useful.
Uh, I told when the level of dopamine goes very high for a long time, it makes very several damage to dopamine receptors and dopamine cells.
Okay. Yeah. Okay. So let me interrupt you again. So drugs, like you take amphetamine, you can indeed get a very strong release of dopamine, and that's going to cause all kinds of adaptations in the circuitry. For example, you'll get increased expression of dynorphin, which will cause lowering of dopamine release subsequently. And so there's a set of adaptations that take place to compensate in a homeostatic like way for the acute effects of the drug. But in the case, what happens is that those effects wear off, so then reverse themselves, usually within a few days or weeks, right? So it's just like a heroin addict. If you stop taking heroin, you go through withdrawal symptoms, and then you experience them for a few days to a week, and they can be unpleasant, but they go away. But what doesn't go away is the liability to relapse to drug taking. So that's still there months or years later. So that's why I've felt for a long time that we need to pull apart these adaptation-like things that are responsible for withdrawal symptoms, these memory-like things that are responsible for a long-term liability to relapse. I hope that's helpful.
Yes, I got my answer. Thank you.
Good, thank you.
Any other questions? Huh?
Well, Roger, thank you very much for your presentation. It was very nice. So just a quick question. I don't want to take the time very much. In your test you did when you
sort of induced a dopamine click on the mice. I saw a dopamine decline after the click. Is that correct? A little dopamine decline. Recall?
I would say no. In general, no. Sometimes yes, but in general, no.
Okay, so maybe I was wrong. Sorry.
No, that's okay. I mean, a whole separate debate in the field is, if you're expecting a reward and you don't get it, then you see these dips in dopamine, and how meaningful are they? Can they be used as negative prediction errors? So there's a lot of interesting stuff about decline in dopamine, but we haven't contributed too much to that.
Yeah, but you were talking about before the click, right? Before the expectancy of this dopamine received, but I'm talking about after. Like after the subject receives the dopamine click, after it receives its reward, I think there was a little decline. I was thinking if this sort of induction of reward were to be lengthened, would this decline be sort of also lengthened, or would it stay the same?
I mean, people have done tasks where they give, like, instead of a reward, they give reward, 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 and you can still see sort of pulses of dopamine increase to each of these rewards, if that helps.
Yeah, thank you very much. Got my answer.
Thank you, Professor. Actually, I have two questions. First, about the role of hippocampal inputs to nucleus accumbens and its role in motivation or learning or other cognitive task depends on emotional task depends on the subregion. As Grace said, the inputs from the ventral hippocampus come to nucleus accumbens, and after that, by a way that we don't know exactly, maybe from ventral pallidum go to VTA and excite dopaminergic neurons from the silent state to active state that you mentioned in tonic release of dopamine. But here, after your talk, I think that maybe we can think about the effect of these inputs on the CI neurons, cholinergic neurons in nucleus accumbens, maybe without affecting the activity of dopamine neurons in VTA. This input can modulate the release of dopamine in nucleus accumbens, and maybe it would be another explanation for these effects. If you wanted to explain it.
Sure.
Okay?
Yeah, I mean, just to add a couple comments. So first of all, I think you're absolutely right. And in fact, we know about projections of non-dopamine cells from VTA directly to cholinergic interneurons in the accumbens. So this is work by Christian Lucia a number of years ago. So there's many pathways by which incoming glutamatergic inputs and GABAergic inputs into the accumbens can modulate cholinergic interneurons and thereby modulate dopamine release. So that's absolutely true. One thing that you also said was referring to the Grace work about some silent dopamine cells. I just wanted to note that whenever we record dopamine cells in awake animals, behaving animals, we never see any silent dopamine cells, in the sense that we identify dopamine cells by making them fire because they express channelrhodopsin, but we never see cells that are identified as dopamine in that way that go quiet and then become active when the animal's more motivated. So we just think that's not correct. We think that just should be replaced as an idea.
Okay. Thank you. What about the direct inputs from ventral hippocampal to nucleus accumbens and the axon terminals on cholinergic interneurons? What about?
That's right. I mean, I don't know too much about specifically the inputs from ventral hippocampus to cholinergic interneurons. I believe there's some work done on inputs to fast spiking interneurons in the accumbens from ventral hippocampus. More generally, I completely agree that's an important pathway, and it's going to be very important in establishing things like place reward associations and conditioned place preference, but I don't understand how it works at all. Sorry.
Thank you. And another question is about the maybe not both. Thank you for your complete explanation for the different aspects of dopamine and dopamine stories in Seattle. As we know, traditionally, it seems that dopamine make motivation for behaviors from the nucleus accumbens, and it shifted to dorsal striatum after repetition, after learning. And it's a theory that maybe nucleus accumbens core is initially engaged in this process, and after learning that behaviors came to dorsal striatum, and as you said, efficiently with it, without using many resources, the actions initiated and maintained. So it seems this story is not completely matched with your explanation, and it's better maybe to explain them in details. Especially, I'm not sure about. I'm thinking about the pattern of ramp in dopamine release in nucleus accumbens that you said when rats approach to the reward, it's a ramping in dopamine release in nucleus accumbens. But before that, you said that DLS also is engaged in motivation with lower time scales. Yeah, what's the correlation between two phenomena in DLS and nucleus accumbens about motivation?
Okay, let me see if I understood your question, and I hope I can provide a satisfying answer. So, for many years we've talked about how these different parts of striatum, these different circuits through striatum, can be involved in different features of behavior. And for example, a long literature suggesting that circuitry through dorsolateral striatum was more involved in habits and sensory motor stuff like that. I think that's true to an extent. I'm sorry. Also, you also mentioned this idea of transfer of learning. So you start out by using circuits through ventral striatum, and then maybe you switch to dorsal medial striatum, and then maybe you switch to dorsolateral striatum. So I don't think those things are wrong, but I've come to find it a bit more useful to think about these different timescales. So if you're performing a sequence of actions that you haven't done very often, you haven't had the same level of opportunity to learn about the individual transitions between each moment in the sequence, right? But as you do it again and again, you can learn about those individual transitions, and then you can perform those sequences using a lower level control, right? Whereas you no longer necessarily need that higher level, longer timescale control through the ventral striatum. So that's how I think about it now, that this idea of timescales is sort of organizing many of the phenomena that we've observed in the past. Anyway, that sounds like a longer discussion, and I wish I was there with you to discuss this.
Thank you. Thank you very much.
Thank you, Josh, for the explanation. I would also like to ask two quick questions, and you may feel free to give very brief answers as we are running out of time. So the first question is about the very interesting work you showed at the end about the decoding of the location of the rat. So my first question is that if really that credit assignment, based on the credit assignment story that you said in a model-based system, didn't you expect that more jumps basically? So you showed very infrequent jumps only at the end when the rat was receiving the reward, right? But they were very infrequent. Like a very naive expectation might have been when you get to the reward, then everything in the path back should light up and then come back, in the place. So, but you didn't see that. You only saw a sporadic lighting up. First, second, have you ever gone back and checked those particular locations that lit up? Are those the ones that actually got value update, or does the value update goes to everything? These two small collections. And I give another question for the beginning, the starting part. So in the ventral striatum, you showed ventral striatum to be have a very long time constant. Very interesting. But I guess to prove that ventral striatum dopamine release is still encodes RPE, what can be done is that if you look at a sequence of 0% or a sequence of 75%, which can happen randomly in your data, can let's say a sequence of 0% clicks get the release of dopamine in ventral striatum below the baseline, so you get that negative prediction error that you should get if really it's about a time constant that is long. So these are the two questions, beginning and the end.
All right, let me deal with the last question first, if I still remember it. So first of all, were you a reviewer on our manuscript? Because I have a similar issue I have to deal with. So what actually happens, we think, is that, yeah, the ventral striatum is just taking longer to discriminate. And that if you keep on training, then eventually the ventral striatum would show a dip to the 0% Q. And we're just confirming that right now. That's certainly what happens in the model, but we're still confirming that with extended training. As far as the hippocampal jumps are concerned, so yeah, you're absolutely right. So one thing I had to show this movie because I thought it was cool, but I've only had the movie for a week. So we are planning to exactly ask this question of, are the places that you jump to exactly the places that get value updates, as measured both by subsequent dopamine dynamics on subsequent runs and by the choices the animal makes. So we're definitely doing that. The question of whether the jumps seem less than you'd expect. Well, I only showed you one video, and sometimes you see other trajectories through the maze, but I don't think it's that different to what you'd expect, because what we know is that these so first of all, just to reiterate, there's two kinds of non-local decoding going on here. So one is, as the animal gets a reward, then there's a few seconds where he's more likely to enter a ripple state. And in those ripples, you play back specific trajectories. But you don't get 50 ripples on every trial. You only get a few episodes of ripples. So there aren't that many opportunities to undergo these non-local decodes and replay trajectories. So that ties into your other question. So now we need to go and see if those updates are exactly the ones that are being used. The other form of decoding, non-local representation that's going forward, is this sweeping ahead that you also saw a little bit as the animal's running forward. And what we also want to see is, does the sweep ahead go far enough to reach like representations that the animal would then say, oh, that's a valuable place, I'm going to go that way in half a second from now. So those are both very important questions that we're going to hopefully get to the bottom of with more data analysis. But thank you for the question.
Very nice, very nice. Thank you so much. I would like to thank the speaker once more for his.
Thank you all for your patience for staying for a long day.
Yes, thank you so much.